If you're on Twitter, follow me, because when you leave and you're finished with this course, I still want to be able to reach out and influence you, okay? It's cheap, it doesn't cost anything, and you'll see my virological teachings as long as you're there, all right? I think that would be really persistence, right? All right, when we mention evolution, I at least think of Charles Darwin. And this is a page from one of his notebooks. Charles Darwin, of course, a huge driver in our understanding of how evolution works, how organisms on this planet evolve by natural selection from their ancestors. And I like this notebook because he drew a phylogenetic tree here. And this, of course, I've shown you in terms of nucleic acid sequences. We can arrange viruses in phylogenetic trees where we have ancestors leading to evolved viruses. I think this is cool because he wrote, I think, and he wrote the tree here. But today, we really understand what this process is about. So we can actually take it down to the protein level, because Darwin, of course, studied the evolution of animals and didn't really understand how this selection worked. But now we know that we can have in an organism a host protein which is very well adapted to its environment, to its function, but something can change. You can have a change in the environment or some other factor that now makes that protein maladapted to the environment. However, because all organisms undergo mutations and make proteins that are different from time to time, a new protein may be selected that is now adapted to this new environment. And that will then, if that newly selected protein is well suited for the new environment, it will be selected for over all the other variants that may be present. We call that purifying selection. So this is how we understand that evolution occurs in multicellular organisms, in unicellular organisms, and it happens the same way in viruses. In fact, Darwin would have loved viruses. He didn't know about them, of course. He worked before they were discovered and understood as entities. But today, viruses are the ultimate expression of evolution. There's nothing better to look at experimentally and understand evolution as it's happening. In fact, if you study an RNA virus, evolution occurs so quickly that you can see it occurring in real time. It can happen in a day in the laboratory. So for Darwin, this would have been perfect. It would have made everything very clear, because you can do incredible experiments with viral evolution that you just can't do in another animal. Okay? So today we're going to talk about viral evolution. It's a really big topic for one lecture, but it will give you a taste of where this field is. We define it as the constant change of a viral population in the face of selection pressures. So viruses are always changing, as you will see, and there are always selections on them to select for better fit entities. So in general, when we talk about virus evolution, we like to talk about where viruses came from, where they started and how they've changed, and then from the point of today, where they're going. Prediction is very difficult, as you know, but we have now incredible tools that really teach us how viruses evolve, and so we can make some assumptions about where they might be going. And so, as I said earlier, virology has provided incredible insight into how organisms evolve. And much of our evolutionary theory has been changed because of what we've learned from viruses. So, for example, we have viruses, of course, need hosts. They're obligate intracellular parasites. As host populations grow, different organisms and so forth, virus populations get selected that can infect them. Viruses change hosts often and infect new hosts. And in addition, viruses can select host populations. They have wiped out entire organisms, and they can modulate how an organism evolves itself. And we'll see later how the virus-host conflict, as we call it, shapes the immune system. And if a host population can't adapt, it'll get wiped out. And we being here today, humans, we haven't been on the earth that long, our presence shows that we've been able to adapt to virus infections at least until this point. Now, the public is always confronted with virus evolution, whether they believe it or not, because viruses change in ways that make very public occurrences. We have new viral diseases emerging like AIDS, West Nile, HCV, Ebola virus. These were not present at some previous point in time. We have new influenza viruses arising every year. We have a variety of common cold viruses. Drug resistance is an example of evolution. So people really don't understand this. When viruses change, when they emerge, when they become resistant, this is evolution at work. And it happens every day right in front of you. There are 4 drivers of virus evolution: making a lot of progeny, making a lot of mutants, what we call quasi-species effects. We'll talk about what that is in a moment, and selection. So these are the 4 parameters that make evolution work, and needless to say, you should know what they are and what they mean. They're very important. We'll talk about them today. You should already know by now that viruses make large numbers. This is one of the approaches that viruses have to being successful. Make lots of particles. Many of those particles never get to infect the cell, but a few might, and that virus can continue to see another day. If they made fewer particles, this strategy just wouldn't work. So this is a table with 2 different virus infections on it, showing you the half-life of the virus in the blood, which is the time after which 50% of the viruses have lost infectivity. You can see it's an incredible number for HIV. 6 hours. Every 6 hours, half of the viruses turn over and they're replaced. For hepatitis B virus, it's about a day. Turnover here, 50%, 90%. And the total production in the blood, we talked last time about all the HIV in the blood. Over 10 to the 9th particles a day are produced in the blood of HIV, over 10 to the 11th of hepatitis B virus. So remember, every time a virus replicates, whether it be an RNA or a DNA virus, mutations are produced in the genome. And if there is some selection applied to those viruses, you can have emergence of a new population. So if you're making antibodies or cytotoxic T cells against the virus, perhaps there are a few viruses that aren't recognized by those antibodies. Those will proliferate, spread to the next host, and you will have selected a new population. So the interface of defense and replication is where a lot of selection and evolution of viruses occurs. So large numbers of progenies. We touched on why this happened, why a lot of mutations occur last time when we're talking about antivirals and the problem of antiviral resistance. And this is an important factor for evolution as well. Evolution only occurs when mutations take place. And that's true not just for viruses, but it's true for us as well. If we didn't undergo mutations in our DNA, we wouldn't evolve either, as we have. It takes a lot longer for us to do it, of course. It takes thousands of years for a property to be fixed in the human population, and that's why studying organisms like us is very difficult. But viruses evolve in a day, and so this has really clarified, as I said, our understanding of how selection works. The mutations that are produced, whether they be in us or in viruses, are produced during copying of the nucleic acid. Every time a nucleic acid is replicated, a mutation occurs. Viruses are always mutating. Remember that. If you remember like 5 things, I'm giving myself room here. If you remember 5 things that I've said, and I think I've said this before, another one should be viral genomes are always mutating. And that's why headlines like this one, which is in Time magazine, drive me crazy. And you can just search for the term on Google. Search for viruses mutating. And you will get headlines like this, which is Ebola is mutating, flu is mutating, herpes is mutating. It just is ridiculous. It's not news. They're always mutating. So this one, they're saying Ebola virus is mutating. And this is Time magazine. They really, well, I guess, what the hell, they shouldn't know any better from anyone else. But scientists at a French research institute said Ebola virus has mutated. and they're studying whether it may be more contagious. So it's a fair thing to say whether there's a phenotypic change in a virus or not, whether it's more contagious or resistant to something, but to say it's mutated is absurd. This drives me crazy because they're always mutating, and I think they don't understand. So I want you to understand this and never forget it. Viruses are always mutating. It's not a big deal. Whether there is a phenotypic consequence is really the issue. So I think either the journalists don't understand, or they don't understand that mutation applies to nucleic acids, not phenotypes. Okay. By the way, next week I get to teach a science journalism class here at Columbia. I was invited by the professor to talk about viruses, and you can bet that I'm going to mention this as well as many of the other things that I complain about. Okay. So as you know, RNA viruses, I'll be nice though, okay? I'll be nice. But they're just beginning their careers, so they can be influenced. So last time we talked about how all nucleic acid polymerases make mistakes. Remember that. But the RNA virus polymerases don't correct them. So they have high misincorporation frequencies, 1 per 1,000 or 10,000 nucleotides polymerized. And so in a 10 kilobase virus genome, 1 in 10 to the 4, you get 1 mutation per genome. Every time it's copied, you get a single mutation. The DNA viruses have error-correcting mechanisms, as we talked about last time, so in the form of proofreading. So their mutation rate is lower. They make fewer mutants than do RNA viruses, but they still make mutations. DNA viruses certainly mutate. They just do so at a lower frequency, and therefore DNA viruses are in general different. In my view, RNA viruses can occupy more niches. They cause more variety of different diseases, in particular more acute diseases than DNA viruses, and that may be related to the difference in mutation frequency. Now this graph displays this fact in a nice way. This is a graph of genome size on the x-axis versus the mutation rate. This is the mutation rate in terms of nucleotides per cell or per replication cycle. So here at the lower right, these gray spheres, this is the mutation rate of bacteria. So 10 to the minus 10th, it's quite low, but they do make mistakes, and of course bacteria can be selected in a variety of ways, antimicrobial resistance and virulence and so forth. And then we have double-stranded DNA viruses here in the open circles. We have single-stranded DNA viruses in the blue, and then the others are all sorts of RNA viruses, single-stranded, double-stranded single-stranded of various polarities, double-stranded and retroviruses. And then up here in the upper left are viroids, which are very interesting RNA molecules that don't encode any proteins. We'll talk about that next week. So you can see the RNA viruses have a higher mutation rate than the double-stranded DNA viruses. Here you're in around 10 to the minus 4, 10 to the minus 5. The DNA viruses 10 to the minus 6 to 10 to the minus 7th. But look at this, is very interesting. The single-stranded DNA viruses actually are a little more mutagenic than the double-stranded DNA virus. They're closer than to the RNA viruses. We don't quite understand why that is, because they do have a error-correcting polymerase. They, for the most part, utilize the polymerase in the host cell. So this is an interesting mystery. Anyway, so that is one of the reasons why RNA viruses are the masters of mutation. Now, along with this, it was discovered in the 1970s, 1976 or 78, that because of this high mutation rate, viruses are not just a single sequence. If I give you a tube of poliovirus, not every virus in that tube has the same nucleotide sequence. And in fact, we used to think we were all of one sequence, right? But we're mosaics, just like viruses are. Every cell in my body and your body has a different nucleotide sequence, because as the cells divide, mutations occur. Viruses are way above that. They exist in these quasi-species, and it was first discovered for an RNA bacteriophage called Q beta. And this is a quote from the paper where they described it, which I think describes it beautifully. A phage population is in a dynamic equilibrium with viral mutants arising at a high rate and being strongly selected against on the other. The genome of the phage cannot be described as a defined unique structure, but rather as a weighted average of a large number of different individual sequences. So 1978, it took about 20 or 30 years for virologists to really appreciate what this meant. So any virus population, mostly RNA viruses, but this also happens for DNA viruses as well to a lesser extent. Any population is a mixture of different sequences. So we call that a quasi-species because, the original idea was that a species was an organism of one sequence. Even though that's not true anymore, the name has stuck. So quasi-species. So here it is illustrated. Now on the left is a picture of a handful of 20 or 30 viral genomes in a stock of an RNA virus. And the genome is a line, and the differences in the bases are shown as different symbols. And there are just a few shown because we can't show thousands of thousands. But basically, every genome is different in this quasi-species. So for every RNA virus, this is probably what their sequence looks like. So a virus does not exist as a single sequence as shown on the right. This happens to be the sequence of poliovirus. It doesn't exist that way. When we determined sequences years ago, this is what we would get, but that's because our sequencing wasn't sensitive enough. This is actually what the sequence looks like. So these differences in all the genomes lead to what we call quasi-species effect. When a virus infects a cell or an organism, it's not a single sequence. It is a whole population of viruses that's infecting. And when they replicate in the host, what replicates is the product of all the selective forces of that host, of the host cell. For example, if a virus is transmitted by aerosol, there are forces in the aerosols that select for the most stable viruses. Then the viruses land in the mucosal layer. There are inhibitors there that select out a lot of viruses. Only the ones with the right mutation survive. So many, many levels of selection on this population. And so what you get out are a selected population of this quasi-species that can now go on and to infect a new host. And when they're outside the host, they will be subjected to additional forces as well. This is one of the main reasons why evolution occurs so quickly, because you infect the host with a huge array of different virus sequences, this quasi-species, so you immediately have the ability to evolve within a single host. You select for whatever is good for the moment, and then that moves out and infects another host. One thing you have to understand, which is really important, evolution selects for the moment. It selects the best virus in a host at that time. It's not selecting something that's going to be good in 10 years, or it's not building towards this perfect virus. They go on a minute-by-minute basis. Whatever works is selected for, and that may go on, but if it doesn't, something else will be selected for. So I like to call this the myth of consensus genome sequences. So here again is this poliovirus sequence. I actually figured this out in 1980. This was one of the projects I was doing, and the technology we had at the time gave us a single sequence, a single genome sequence of poliovirus. That's because it's an average of this quasi-species. If you have, a different sequence in every virus particle, there's no way this old technology could determine that. And so this is really an average of the population. Now we know that with deep sequencing, next generation sequencing methods, we can actually start to look at the quasi-species. We can take a tube of poliovirus and know every individual genome in that population. All right, this is not easy because the sequencing methods themselves introduce mutations, and we don't care about those, right? But people have worked out ways to ask, what is the composition of the quasi-species? And of course, it doesn't look like anything like this. So this sequence that I made of poliovirus probably doesn't even exist in any tube of poliovirus anywhere. Here's an interesting experiment which we do every day. We take this sequence as a DNA and we make an RNA from it. So that RNA has pretty much one sequence. We put it into cells, it starts to replicate. By the time the viruses come out, we have a quasi-species. Every virus is different. And that's because the RNA polymerase makes tons of mistakes. Many of those aren't infectious, but many, many are. And so you have to think of a virus not as a single sequence, but as a population, and any effects of selection happen on that population. So that's what a quasi-species is. It's a little bit abstract maybe to wrap your head around, but it's very important. So it accounts for a lot of the variability of viruses, but there are two other processes that are also important. They contribute to the variability of the quasi-species, and we've talked about these before. These are recombination and reassortment. So again, when an RNA molecule is being copied in a virus-infected cell, here's a polymerase copying this donor molecule. The RNA polymerase can switch templates, and so now this one has jumped across to the blue template, and the result is a recombinant molecule. So this happens often, and with a quasi-species, you can imagine this would generate even more variability in the virus population. Some viruses don't do recombination, they do reassortment. They have segmented genomes like that of influenza virus, so if two different influenza viruses infect the cell, the segments will all be mixed up, and the viruses that come out can have segments from both parents. So again, these viruses exist as quasi-species, and the reassortment amplifies the variability on top of that. So now we take this incredibly varied population of viruses and we submit it to selection. Every time a virus passes through the air, sits on a table, infects someone, it's subjected to various selections. It could be sunny out, so you're selecting for the most thermostable viruses. It lands on a mucosal surface. You select for the viruses that are resistant to antibodies there or whatever antimicrobial compounds might be present. So you end up selecting what we call survival of the fittest. A rare genome with a particular mutation survives a selection event. However, all the other mutations on the genome which don't impact that selection property get carried along as well. So if there is a single amino acid change in the capsid that lets a virus be stable on this table long enough so it can be transmitted to someone else, that mutation will persist. But all the other ones that don't impact thermostability will also come along for the ride. So we call that survival of the survivors. The unlinked mutations get a free ride. So every time a virus infects a new host, you get a new diverse population produced from this quasi-species. Now, I don't want you to think that mutation rates are a bad thing. Of course, they're bad in some cases. If they lead to cancers in us, that's not good. But as far as evolution has occurred in nature, diversity is actually selected for. You might ask, why don't the RNA polymerases get with it and stop making mistakes? Well, we can actually make RNA polymerases in the laboratory that are less error prone. It's easy to do. So here's on the upper right is the poliovirus RNA polymerase, replicates the RNA genome of polio. A number of years ago, two different groups found that a single amino acid change at position 64 here, glycine 64, this happens to be very near the palm domain, so the palm is where the polymerization occurs. This single amino acid change makes the polymerase make fewer errors. It improves its fidelity. And you can make a virus with this RNA polymerase in it, and the virus grows pretty well. But if you make a competition with this virus and a wild-type virus, the more faithful polymerase will lose every time. So in scenarios of competition, being more faithful to the template loses out. And if you put this virus in an animal, it's not pathogenic. In fact, people are exploring these kinds of mutations as vaccine candidates. So making fewer errors makes you unable to compete with wild-type viruses and unable to cause disease in an animal. So the end result of this is that high error rates are actually selected for in nature, in both viruses and bacteria. Similar experiments have been done in bacteria. You can make bacteria with more faithful DNA polymerases, and they don't win. They always lose. So we need mutations. They have been selected for in nature. If they hadn't been, we would have all the RNA polymerases having this glycine, which makes fewer errors. So making mistakes is good, and it's selected for. Just to fill out this story, in this viral RNA polymerase, if you make a single amino acid change here at histidine 273, that mutation, that change in the polymerase makes the polymerase make more errors Okay, so you can either have it make less errors or more. Having more errors isn't good either. So the virus has evolved to be just at the right point where it's making just the right number of mutations. This virus that makes more mutations, again, cannot compete with wild-type virus and is also attenuated in animals. It doesn't cause disease. Now, why am I mentioning this animal business? Well, to infect an animal, you have to go through lots of selection. It's not like a dish of cells where you put virus on and the virus, oh, look at all these susceptible cells I can replicate in. It's great. In an animal, it's to go through several stages. It's got to get through mucosal barriers or spread through the blood or invade organs. So there are lots of points of selection, and that's why having a variable polymerase is really important. So the key here is that diversity is selected. So this relates to a concept, the last experiment I told you where you make the polymerase more mutagenic. This relates to a concept called error threshold. Okay, this is the point at which if you make more errors over this error threshold, the virus is no longer infectious. So mutation is great, but it has to be balanced. You can't make too many mutations, and the limit is error threshold. If you go over it, the virus loses infectivity. If you go too far below it, you cannot make enough mutations to survive under selective forces. So the poliovirus mutation that reduces the mutation frequency, the virus cannot survive. Turns out that RNA viruses exist very close to the error threshold, whereas DNA viruses, as you might predict, are pretty far below it. They're obviously at a place where they can exist. They make enough mutations to be competitive, but they're below the error threshold. And I want to show you why we think this is by two different experiments. Okay, error threshold for DNA viruses. So you infect a cell with a DNA virus in the presence of a base analog. In this case, it's called 5-azacitidine. Now, this is a drug that looks like a triphosphate, but it will not template properly. So it's incorporated as a C, but the polymerase thinks it's a T. So normally if it were a C, the polymerase would put a G in, right? But the polymerase says, oh, this is a T, so it puts an A in wherever it sees an azacitidine in the genome. So basically it's a way of introducing mutations into the genome. You're changing what should be a G to an A. So if you do this with a DNA virus, you increase the mutation rate, but the DNA virus doesn't suffer. Okay, that's because the DNA virus is existing way below its error threshold. You can introduce lots and lots of mutations into a DNA virus genome by a procedure such as this, but it won't lose its infectivity. Now, if you do a similar experiment with an RNA virus, you can only go up 1 or 2-fold in error frequency, and then the virus loses its infectivity. So you cannot make more mutations in a significant rate. And this is that experiment with an RNA virus. So here we're taking poliovirus, and we're treating it with an antiviral, actually, called ribavirin, which up until this experiment, people didn't know how it worked. It was known that this was a nucleoside analog. It was incorporated into the RNA, but it wasn't clear what it did. Turns out that it's a mutagen. When this ribavirin, sort of like the 5-aza-C, when it's incorporated into the genome, it makes mutations. So here is a graph of the specific infectivity of the RNA genome, and they've used increasing concentrations of ribavirin in this experiment, which ends up introducing more and more mutations into the genome. So here is untreated virus here, and then right away, if you introduce 2 mutations into the viral genome by the use of ribavirin, look at how the infectivity plummets. It's already down to about 40%. Between 6 and 8 mutations now, you're almost at 0. All right? So just a handful of mutations introduced into this RNA virus genome almost completely abolishes its infectivity. In contrast, you can put many, many more mutations into a DNA virus genome. It doesn't affect its infectivity. So again, the conclusion is that DNA viruses are existing way below their error threshold. That's probably because they simply can't make that many mutations, whereas the RNA viruses take full advantage of their error-prone polymerases. They're existing right at the threshold. This is actually a good antiviral approach. People have been trying this with other RNA viruses, including HIV. They're trying to develop antivirals like ribavirin that mutate the genome, so it will no longer be infectious, but it will replicate and provide antigen to provide an immune response and be protective. Okay. So also related to this whole idea of diversity, quasi-species and error threshold, there's another very interesting concept which also illustrates the importance of diversity, and this is called genetic bottlenecks. So in this experiment, what we have done, we've plaqued out an RNA virus. Remember the plaque assay? You can do this. You do this under a semi-solid agar overlay. You can pick an individual plaque, and then you expand that plaque, and you grow it up, and then you do another plaque assay. You pick another plaque, and you just keep doing this many, many times over. All right, and 30, 40 times. So you're going from plaque to plaque, and after about 20 or 30 times of doing this, you end up with a virus that can barely grow. You start out with a nicely growing virus, it can barely grow. So why is this? Everything has been constant. All you've been doing is plaquing it. It's the same cell. It's not like it's the same cell you grow this virus in, so you're not changing the environment, you're not changing the medium. All you're doing is plaquing it from cell to cell, from plaque to plaque. But why is the fitness going away? Well, the answer lies in the idea of a genetic bottleneck. What we're doing is by picking a single plaque, we're only transferring a small number of virus particles from one culture to another. And maybe a few thousand are in a single plaque as opposed to many, many millions, which would be if you're infecting cells with a liquid stock of virus. And so I think you know what a bottleneck is. Here you have, to illustrate it, a bottle with this parent population of viruses, but the neck restricts the ability of the viruses to get out. So what you have are just a few viruses that you use to infect the next cell. So think of that single plaque, a few thousand viruses. We're going from plaque to plaque, and that's why the fitness is going down. And the drop in fitness is caused by a known phenomenon. This has been known before viruses were really understood, called Muller's ratchet. And this is a genetic theory that applies to organisms that reproduce without having sex, which do binary division, for example. So sex has been thought to evolve to introduce variability, in particular to provide resistance to infections. But asexual organisms, when they replicate, when they divide and low numbers are involved, they don't have a great deal of genetic diversity. So RNA viruses, which are already close to their error threshold, as I've shown you, what we are doing is restricting the diversity of the population by simply picking a plaque and going from cell to cell. At the same time, mutations are accumulating because of the quasi-species, but there isn't enough diversity to rescue those mutations. And so that's why our fitness decreases. So this whole idea of a ratchet, here is a ratchet in case you didn't know what a ratchet looks like. The metaphor means each mutation is introduced in the genome and it can't be removed. You can't go back, so the mutational gear moves forward but not backward, and eventually these mutations accumulate and the fitness of the virus goes down. So a key component here is we're passaging very small numbers of viruses, so we have restricted diversity, because remember, diversity is selected for, but we are restricting it artificially. It's sort of like making a polymerase that makes fewer errors. We're just using fewer viruses. And then as we grow the virus, the mutations are accumulating in the genome. They can't be removed. I'll tell you how you remove mutations in a moment. And they're fixed by this ratchet metaphor, and therefore the viruses lose infectivity. So here's an illustration of this using our quasi-species illustration. So here on the left is a nice, diverse quasi-species. So let's say this is a virus population, and we take 1 million of these viruses and we infect a new cell. We're getting a big diversity of mutations. If there's some selection in the cell we infect, we've got plenty of genetic diversity to pull one virus out that works particularly well in that. And this quasi-species is simply maintained from cell to cell as long as we inoculate the cell with a lot of viruses. When we pick a plaque, we're restricting the diversity of the quasi-species. So for example, let's say we pick a plaque and all the viruses look like this one in the middle, star, star, triangle. Now, these 3 mutations will all be present in the new genomes, in addition to some others that arise. And you can see how restricted genetically these are right away, because you've used such a small inoculum, and you keep repeating this. You repeat passage of individual quasi-species, mutations accumulate, and because there isn't diversity, this virus loses infectivity. So that's the reason why diversity is important. And this actually happens with quite a number of viruses. These are some experiments where bottleneck passages, the same kind I've shown you, plaque to plaque, have been carried out for different numbers of passages. This is a bacteriophage, 2 bacteriophages and a variety of animal viruses with different passages. And you can see here the percent average decrease in fitness. So it varies per virus, but it's a universal property of RNA viruses that if you use a restricted number of viruses to initiate an infection eventually, if you do that over and over, you're going to lose fitness because of this requirement for diversity. So does this happen in the real world? You may be asking, okay, this is great, but in real infections, is there any kind of situation where diversity is restricted? And the answer is yes, of course. For example, when you're infected by influenza virus, you may get a droplet that has a few thousand viruses in it. And then you may pass on droplets to someone else with just a few thousand viruses in it, right? So you can have a serial chain of transmission, which is bottlenecking. Moving a virus by insect bites, where the bite is transfers a very small amount of liquid and there's very little virus in it. Or even, when herpes virus is reactivated in your peripheral ganglia, probably not much virus is produced, and that is spread to another person by secretions. That is also a bottleneck situation. So there are examples of potentially bottlenecking situations in the real world, but obviously viruses transmit effectively. So there must be a way to rescue a virus from Muller's ratchet, so to speak. And in fact, there are ways. Now, in the laboratory, remember, we're doing this plaque-to-plaque passage, which is not good for virus fitness. If instead of taking 1 plaque, we took 10 and pooled them and then passed them and took 10 more plaques and pooled them, we would not have a bottleneck, because we're using enough virus inoculum and we're providing enough diversity so that any deleterious mutations will not predominate in the virus. But in nature, we can't control the inoculum, of course. So what happens is the disadvantages of having a small diversity, those are overcome by recombination or reassortment. So viruses recombine with one another to eliminate deleterious mutations, or they reassort fragments. And this, of course, happens randomly, but the one virus that is infectious and able to survive under whatever the conditions are then go on. So you don't have Muller's ratchet in nature because of this. So here's an example of that. We've generated 2 viruses which have deleterious mutations in them because you've been inoculated with a small amount of virus, and these mutations have arisen. But these 2 viruses can undergo recombination to make a recombinant which now has rescued both of those mutations. So this is a simplification of it, because recombination is probably quite extensive. But this is just one way, in addition to reassortment, how in nature Muller's ratchet is overcome by simply providing more diversity. So this is really providing additional support for the idea that the quasi-species is really important for virus survival. Diversity is essential, and if you remove diversity either by Muller's ratchet type of experiment or by reducing the fidelity of the polymerase, the population suffers and it can't survive. And that's why this property of high mutation rate has been maintained in all the eons of virus evolution. Let's look at a couple of examples now, concrete real-world examples where I can show you how mutations arise in a genome and how they are selected for in real-life situations. And there are 2 different kinds that we're going to talk about. They're called genetic drift, and these are essentially single mutations in a viral genome that arise by errors in the polymerase that we've talked about. And the other is genetic shift, and this is diversity that arises by recombination or reassortment. Now, you can imagine one of you gets infected with influenza virus, and the virus is replicating in you. It's producing these huge quasi-species with all sorts of mutations in the genome, and you're making an antibody. Maybe you've gotten vaccinated, and you make a quick antibody response, and most of the viruses are neutralized. But maybe there's one virus in you that has a mutation that leads to a change in the HA, hemagglutinin, that doesn't allow the antibody to bind. That virus is going to grow pretty well in you, and even though you may not get sick, you'll probably transmit it to someone else. You may transmit a bigger inoculum of that virus to someone else. So eventually that virus will spread in the population. And that's what happens with influenza virus from year to year. That's genetic drift. So the immune system is a really good example of selection, because it's quite clear that antibodies are cytotoxic T cells which recognize viral peptides in MHC molecules. The viral peptides can change. The epitopes recognized by antibodies can change as well. So you should know all about influenza virus by now. We've talked about it quite a bit. It is the HA and the NA glycoproteins on the surface that determine whether it's A, B, or C influenza. And within the influenza A viruses, we further classify them into subtypes depending on the type of the hemagglutinin and the neuraminidase. So far we have identified 18 different hemagglutinins and 11 different neuraminases. So an H1 hemagglutinin, you can make antibodies against it, but those antibodies will not block infection with a virus with an H2 or an H3 hemagglutinin and so forth, and the same with the neuraminidase. Even within the H1 hemagglutinin, there can be drift from year to year, single amino acid changes, such that the antibodies you produce one year will not block infectivity with that virus in the next year. The influenza viruses we worry about are the H1, H2, and H3. These are the human influenza virus strains. They transmit well among humans. All the others, up to 17, can infect birds of all different species, water birds of different sorts. And the 18th hemagglutinin was isolated from a bat. Now you may wonder where the H5 hemagglutinin is in this listing. Well, viruses with H5 hemagglutinins can infect birds. They periodically jump into people and cause serious illness, but they haven't yet transmitted among people. They're not human influenza viruses, so that's why I don't include them here. So here's an example of genetic shift in influenza viruses. This is one of the great examples we have of this. Periodically, brand new influenza virus strains emerge that have a new hemagglutinin and or neuraminidase. So the population is not immune to this virus, and the virus spreads globally. It causes a pandemic. Millions and millions of people get infected. There can be lots of mortality associated with it. So one of the most famous pandemics is 1918, which killed many millions of people globally. It is believed that this virus actually originated in a bird species of some sort, possibly a duck. And so all of these RNA segments came from this avian source. But this is a little difficult to prove because it was before we were isolating virus. And of course, we have no ducks left from that era anyway. Now in 1957, this virus circulated for many years, and every year it changed a little bit. So the flu vaccine didn't come on the market till the 40s, but shortly after it was introduced, a few years later, it didn't work anymore, and people wondered why, and that's when drift was discovered. So this virus drifted slowly in between those years. 1957, a brand new virus emerged with totally different hemagglutinin and neuraminidase, H2N2. This caused a global pandemic of influenza virus, and this virus was a reassortant. So the red RNA segments came from the 1918 strain, but look, these yellow ones, one of the polymerase RNAs and then the HA and the NA, came from an H2N2 virus from ducks. So these viruses reassort in animals in the wild, and when the right reassortant occurs that can infect people, boom, somehow it gets into people and you have a pandemic. The next one was 1968, the H3N2 virus emerged. You can see it has a few of the segments from the original 1918 that have been passed along. It has a single one from the 1957, the NA, and then 2 new segments from a duck virus, which again donated these segments in some animal. We don't know where the reassortment occurs. We suspect that it's pigs. But this is an example of the variation generated by reassortment. Now, in 2009, a new pandemic strain emerged. It was an H1N1 strain similar to the 1918 virus. In fact, if you had lived in 1918 and been infected with that virus and survived, those old folk were actually immune to infection with this virus. The reason why this virus emerged was that in 1918, that virus, when it infected people, also infected pigs. And in pigs, there's much less genetic variation of influenza virus. So that virus has been maintained in the pig lineage all those years. And then in 2009, it donated some genes to a reassortment and jumped back into people. So a lot of the problems with influenza are really coming from our increased growth of pigs for human food and also of birds, chickens and so forth for food production. So this virus has a very interesting origin. It is believed to have originated from several different viruses, including a Eurasian swine virus. So there are lineages of pigs, and they have specific influenza viruses in them. The classic swine H1N1, that's the one that stayed in pigs from 1918. A human H3N2, which emerged in 1968, and an avian virus. And these formed an intermediate reassortment, which eventually gave rise to the 2009 strain. You can see it derives RNAs from all these different viruses. You can just look at the colors and tell that. And we suspect that this reassortment occurs in pigs because pigs can be readily infected by viruses excreted by birds. In birds, interestingly, influenza is a gastrointestinal infection. It's not a respiratory infection. It passes through their GI tract, and they actually excrete the virus as they're flying over pig pens, and the pigs get infected and so forth. So we think this happens in pigs, and then people working with pigs on pig farms pick up the virus, and then they spread it to the rest of the world. But nice example of selection. So here we have a population that's immune to an H3N2 virus and the previous H1N1 that had been circulating. All of a sudden, this brand new H1N1 comes in. Nobody has antibodies except the 1918 survivors, and boom, you have a big pandemic. So that's the selective force. The antibodies select for the emergence of this new virus, and then it spreads through the population. All right, so that's antigenic shift. It's big changes mediated by reassortment. Brand new HAs and NAs get in. The other kind of selection, real-world selection, so you can actually see what's happening, is antigenic drift of influenza virus. This happens year to year as a single virus, H1N1 now, or H3N2, are passing through the population. The pressure, the immune pressure put on by the host selects for variants that can evade the immunity. And in the simplest example, it's if you have antibodies against an H1N1, let's say you've been immunized last year, when you get infected, the variants in you that can evade antibody neutralization will predominate and spread to others. So this is the influenza virus. Again, of course, the HA is in blue and the NA is in red. And just to remind you, that's your ion channel right there through which the protons need to go and which is blocked by amantadine. Here's a model of the HA on the right. It has a globular head that is important for attaching to cell receptors in a fibrous domain, and then it's embedded in the viral membrane. So these colored areas are the antigenic sites on the top of this protein that vary. And these are the sites that bind antibodies. If antibodies bind any of these, they will block infectivity. But you can have a single amino acid change in one of these sites, and the antibody will no longer bind. So you will get a mild case of influenza, and then that next year the vaccine will have to be changed to accommodate that. So this is drift, small changes from year to year, and they make us change the vaccine periodically. But again, they arise because as the virus replicates, it makes lots of mutations. Some of those may encode changes in these antigenic sites. Remember, they're all occurring randomly. Billions and millions of mutations. And if you have antibody in a person present against one of these, will eliminate the virus, but if there's a mutation that escapes the antibody, that's the selection that that virus needs to proceed. So that's what we mean by selective forces governing the evolution of this virus. Now, the interaction of the immune response with viruses has been able to be characterized at incredible detail in the past few years, because we can sequence not only viruses, we can do deep sequencing of many, many virus isolates, but we can sequence all the animal genomes that are out there. And you can look at the sequences of individual proteins. So a concept has arisen because of these data called the host virus arms race. And basically what this means is that you have a host protein. This could be an antibody, or it could be any host antiviral protein that neutralizes a virus. And eventually the virus will alter to change the way it interacts with this protein, so it escapes. The virus is winning, we say. But hosts change as well. They change more slowly. With time, and we're talking about a long time, the host can evolve to have a mutation in this protein that will now accommodate the virus mutation, and now the host will win. So we thought this was a possibility for many years, but being able to sequence genomes of viruses and their hosts has made us actually see this. So we call this host virus arms race, and they're called Red Queen conflicts. All right, there's your Red Queen. Remember her from Alice in Wonderland? She said, I have to do all the running I can just to stay in place. And that's what these virus host arms races do. One mutates, the other mutates, back and forth. In the end, nobody wins, and they're just where they started from. So Red Queen conflicts. Let me show you how you actually study these, because it's quite interesting. It's a brand new field in virology and evolution. So the first part of it is that you can establish phylogenies of species, not based on morphological criteria as Darwin did, but we can take the sequences of us and our ancestors and put them in a phylogenetic tree. And we have sequences of the genomes of all the individual organisms in this phylogeny. Now, when a mutation is selected for in a population, say because a virus selects for that particular defense mutation, we call that positive selection. So you can look in these genomes for, so you can compare human versus chimp versus gorilla, etc. In a particular protein. We call those proteins orthologs. We can look for codons that are positively selected. So here you see these have been arranged in terms of phylogeny. We have a particular codon which seems to vary. So in this particular protein, all of the other amino acids are conserved, but here's a hot spot where over time of evolution, over the phylogenetic years in these different organisms, this amino acid has changed. So we call that positive selection, and the assumption is there must have been some event that selected for it. And of course, if the protein we're looking at is a defense protein, an immune system protein, then it's likely that whatever selected for these changes was a virus or a bacterium. So you do these phylogenies, you look at the protein you're interested in. You can actually look at many different proteins and look for evidence of positive selection. And then you can ask, do these have anything to do with a virus? So then you take cells from each of these species and you put them in culture and you infect with a virus that you think might be involved. And you can ask whether the host is winning or the virus is winning. So if the virus is winning, it can overcome the defense and it will replicate well. If the host is winning, the cells aren't killed by virus infection. So you bring this into a cell culture dish and you can make very simple experiments to test whether these changes have any effects. And then once you establish a virus host cell protein pair in this way, you can then change the amino acids and prove that the changes had an effect. So here, for example, this particular species, the virus is winning. It has an S at this amino acid position. When the host is winning, you have G's there. So you can simply take the protein which has the S and in which in the cell that this protein is existing, the virus replicates quite well. You change the host protein to a G, and now that accommodates the virus change, and the host will win. And by winning, we mean that the virus isn't replicating well in the cell. So this has been done for a variety of viruses and cell proteins. And the cool thing is that this is sampling change over long periods of evolution, because the phylogenetic trees are arranged in accordance with ancestors of the current organisms. And we know, for example, on Earth today, there are organisms that are quite ancient and others who are derived from them. So we're sampling the eons of evolution in terms of protein changes. So here's an example of this, where we have two different host cell proteins that have been studied in this way. One is TRIM5 alpha, which is an antiviral specifically for HIV. And by analyzing these conflicts over time in different species, we can see that there are a number of positively selected amino acids in a very specific part of TRIM5 alpha that have evolved under selection by HIV. So we can look in different species of primates, for example, and see this changing, and that change goes along with susceptibility to infection. Now, many antiviral proteins affect many different viruses. So PKR is one. Remember protein kinase R? This is a interferon-induced gene that phosphorylates the IF2 alpha and shuts down host cell translation. PKR is antiviral for a number of different viruses. And you can do this virus host arms race experiment with PKR in all of these different viruses. And you can map on PKR the positively selected amino acids that have been selected by the different viruses. So for example, here in the RNA binding domain, you can see positively selected residues that have been selected for resistance to herpes infections, influenza, pox viruses. And another part of the protein, the kinase domain, positively selected residues for hepatitis C, another pox virus, HIV. So each virus is interacting with a different part of the protein. The protein changes, then maybe thousands of years later, the virus changes, and then the host cell protein changes again. So that's what we mean by positive selection. These residues are changing over time to accommodate viral changes. So if you look at the protein in 3 dimensions, you have 3 different viruses interacting with 3 different parts of the protein. So these are always changing. And if we do this for the next 10,000 years, presumably these sites will continue to change as well. So that's what the cool part of this is. You can sample what we think happened tens of thousands of years ago simply by looking at the sequences of animals on these phylogenetic trees. So you understand now that there's a lot of diversity among the virus genomes, especially among RNA virus genomes. Yet, for some viruses, that diversity doesn't seem to be expressed very effectively. So for example, I've just told you how flu varies from year to year. But poliovirus, there are only 3 serotypes. There's only 1 measles serotype. Now poliovirus is a icosahedral virus, whereas influenza virus, of course, is envelope. So you could argue that, well, maybe envelope viruses can be more flexible in their changes that are accommodated. But measles is also enveloped in the middle there. 1 serotype of measles. We have been using the same measles vaccine for 50 years. The virus does not change antigenically. So measles virus makes as many mutations as flu. The genome is mutating. It has a quasi-species. And probably there are genes under positive selection for measles virus, but not the ones constituting the structural glycoproteins. So this is actually going back to this idea of viruses mutating. Poliovirus is always mutating, but it's not expressed in terms of the capsid protein. That's why we only have 3 serotypes, and the same polio vaccine we've used for 50 years still works today. Why this is so is an interesting question. What is the reason why poliovirus and measles virus cannot undergo this antigenic change is a puzzle. We really don't understand it, especially in the face of the fact that everything else in the virus is quite plastic. So selection, now you understand selection of mutations under different conditions. You can select for virulence or avirulence, as we've talked about before. So the interesting question that arises is virulence a positive or negative trait for selection? So in other words, you would think that most viruses would evolve to be maximally transmissible and minimally virulent, right? You don't want the virus to kill the host, but you do want it to transmit effectively between them. So according to that theory, all viruses should be highly transmissible and not virulent. So many viruses are not virulent in us, as we've talked about before, but many are very transmissible and also very virulent. So what's going on here? And there's an interesting experiment that was done in the wild a number of years ago in Australia, which illustrates what our expectation of virulence should be. That is, it probably evolves to that viruses are less virulent with time. And this is an experiment done in Australia. So in 1859, the European rabbit was brought to Australia for hunting purposes. Okay, so they had rabbits in Australia, but they were too fast and hunters couldn't shoot them. So they brought in these slow European rabbits, and but within a few years, these guys multiplied like crazy, and they overran the country, and Australia decided they had to get rid of them because they were being pests. This is actually a picture of one area with all these rabbits. So they brought in a virus to do this, myxoma virus, which is a pox virus. It was released into the wild in Australia in the 1950s. The idea was to try and get rid of this rabbit. So here on the right, this is the European rabbit, and on the left is the cottontail, which is native to Australia, but the virus doesn't kill these cottontails, it just causes a rash, whereas this virus kills these animals. And the way the virus is spread from rabbit to rabbit is by mosquitoes. The animals develop, these animals, the cottontails develop warts, but the European rabbits die. So 90 to 90% fatality. So they released this virus thinking it's going to wipe out all of the animals. And the first year the virus killed 99.8%. That was the mortality rate. But in subsequent years, the mortality dropped. And I'm not giving you a number because it's a bit complicated, but let's say that in the end, after about 8 years, the mortality rate was anywhere from 50 to 70%, so significantly lower than the 90%. And eventually the rate of killing dropped below the reproductive rate of the rabbits, so they stopped because it wasn't going to work anymore. And the rabbits are still running around there, and this is a failed experiment. So why did it fail? Because both the virus and the rabbits reproduce prodigiously. We know viruses do, I've already told you that, but rabbits in particular have a high offspring rate, and they do so very quickly. So the virus evolved to kill fewer rabbits. So this is, according to our theory, the virus would evolve to kill fewer rabbits so it could overwinter and spread in the spring. And this has been verified by studying these viruses. They were isolated from the field and inoculated into rabbits in the laboratory. And in fact, the viruses did evolve to lesser virulence. At the same time, the rabbits also evolved as well. Can you consider that first year, 99.8% of the rabbits were killed, but the ones that were left were the mutants that could resist infection, right? Those were the ones that had a protein, a defensive protein that could bind the virus and neutralize it. So the combination of the virus and the rabbits mutating made for this to be a failed experiment in rabbit control, but it illustrates this idea that viruses, initially coming into a new host anyway, are very virulent, but then they evolve to exist in a stasis so that they can persist. Now, I don't know of any evidence of this happening in people. It may have happened a long time ago. Say a new virus enters humans, maybe over time it becomes less virulent, but we haven't been studying viruses long enough to be able to see this. We've only been studying viruses about 100 years, and there's no evidence for any of the current viruses that there's been a reduction in their virulence. Maybe the best examples are recently acquired human viruses. So HIV is an example of that. It went into people about 1920. And, we only detected it around 1980 or so. And there's no evidence that it's reducing in virulence. I mean, you will find some papers here and there that say that it is, but I don't think that's the case. So this is an interesting concept, but I don't see, except for experimental results such as this one or things you can do in the laboratory, I don't see any evidence for it yet. All right, let's end up by talking about where viruses came from. Where did they originate? unfortunately, we don't have a fossil virus to look at. So you can tell all the dinosaurs that were around and how old they are by digging up fossils, but we don't have virus fossils. The oldest virus stocks we have are the 1918 influenza virus and a 30,000-year-old giant virus, which was recovered from the Siberian permafrost. It's dated to be it was actually infectious in the ice, and by the carbon dating of the sediments, it's estimated to be about 30,000 years old. So we can't just go dig in ancient sediments where these dinosaurs are and find viruses and say, there are old viruses. But we have other ways of doing this. Okay, we have some ways to say, can we backdate virus evolution according to the rate of change of contemporary viruses? So if you look at the herpes viruses, you can calculate the evolution of the genome and basically figure out that the three groups were around 180 to 220 million years ago, which would put them around when the dinosaurs were present, as I told you in the very first lecture. So here's the time scale of life on Earth. Here's formation of Earth 4.5 billion years ago. And so here would be 200 million years ago or so. But that's not really that satisfying. It's really nice, but there are actually better ways to back calculate how long viruses have been around. And a really nice way is helped by the fact that there are many virus sequences in genomes of animals. There are endogenous retroviruses, which we talked about. There are also pieces of other viral genomes, not retroviral, but other viral genomes. You can use this to tell when those went into the genome. And this is a field called phylogenomics. So we have all these different animals. You have an ancestral host, and then you have a phylogenetic tree. They're all rabbits here, but they're meant to be different species that have evolved from each other. And you know roughly the age of these animals when they speciated. So for example, these two individuals split around here in time, and this one split earlier. And you can look at where the viral sequences are integrated in the chromosomes and tell about when the integration occurred. So if these two individuals share a common integration site, so the virus DNA has to be in the same place in the chromosome in both species, that means it infected them before they split in speciation. So this integrated endogenous virus element went into the precursor of these two animals somewhere around here. So you can get a minimum and a maximum age. And you can do this extensively for many different sequences that you find. And as I said, this is being made much easier because we can sequence all of these animal genomes, and we can do so very rapidly. Here's an example of this kind of phylogenomics using pieces of single-stranded DNA virus that happen to be integrated into host DNA species. So for example, we've got parvoviruses and circoviruses, and you can tell roughly when these integrated into the genome, 30 million, 60 million, 90 million years ago by the speciation, where they are in the chromosome and whether they occurred at a certain point in phylogeny. So this has told us a lot about when viruses entered. But of course, we're limited to having animals that we recognize in this phylogenetic tree. We suspect, as I said at the very beginning of this course, that viruses originated much earlier than the first animals. In fact, there are two theories about the origins of viruses, and one of them is that they have a precellular origin, that there were replicating molecules in the primordial earth before cellular life emerged, and these became the first viruses. And in fact, that cells emerged from these viruses as well. We don't really know how this would happen. This is a hypothesis, but there is some evidence for this. In fact, you cannot trace the sequences of viruses all the way back then because they have changed too much over time. You can only recognize a virus 200 million years before this, but previous to that the genome has mutated so extensively that we no longer recognize it. So even if we had any samples that were ancient, we wouldn't be able to tell that they were viral. But there are conserved protein structures among viruses that have no sequence homology whatsoever. All right, and this is quite extensive over different kinds of protein structures. So this suggests that there's a very ancient common ancestor to all the viruses. The other theory is that viruses are simply escaped genes from cells. This continues to be proposed, but actually is rather easy to refute, because among the RNA viruses, for example, none of the genes resemble anything in cells except perhaps a small part of the RNA polymerase. So we actually think that viruses, I think that part A is probably right, and these viruses have been around for billions of years. The viruses that infect us today all evolved from animal viruses, because all the viruses that we have were present in animals before humans emerged. So let me give you an example. Pox viruses. Sequence analysis of pox viruses suggests that this is a relatively recent introduction into humans, and probably from a gerbil about 13,000 years ago. And this virus, smallpox virus, is closest to a virus that infects germals. So someone had a gerbil as a pet maybe many years ago. The virus crossed over from the gerbil, and then it spread in the human population. Measles, another similar origin. Measles virus is closely related to a virus that infects cows. It's called rinderpest. And so we think when we began to domesticate cows for human food production, we got close to them. They were not wandering around in the woods anymore, whatever their precursors were. As soon as we started domesticating them, we got infected with them, and then as cities grew in number, the virus then spread. It originated in the Middle East, and then colonization of the Americas brought it here in the 16th century. So basically, new viruses arise from what we have. Brand new ones aren't made. It's simply an evolution of the existing ones, and that brings up an interesting problem. What else could we expect out there? What's the number of all possible mutations that could be introduced and give rise to brand new viruses? Some experiments have shown that you can mutate half of a viral genome and still get infectious viruses. So let's do some math on this. You have a 10 kilobase virus genome, 10,000 bases. There are 4 to the 5,000 different sequences that you could assemble from that 10 kilobase genome. All right, that's just by mutating at every position. 4 to the 5,000, then deletion, recombination, reassortment can add to that as well. How big is that number? There are only 4 to the 135 atoms in the visible universe. It's a huge number. So the possibilities are endless, but we don't see endless viruses emerging. There are some constraints on virus evolution. So for example, an influenza virus is always going to probably look like an influenza virus. It's not going to change to get an icosahedral capsid. A DNA virus will probably always remain a DNA virus, and so forth. So there are constraints on genomes that restrict the kinds of evolution that can take place. So the genome is a constraint, as I've said. The replication strategy probably can't change. The nature of the capsid, selection during infection probably restricts evolution as well. So although there are seemingly infinite possibilities for the number of different mutants that a virus could spawn, in reality they're probably limited. And although there are lots of viruses on Earth, that probably limits the kinds that are going to emerge in the future. Now let me leave you with one interesting fact. It took 8 million years for chimps to evolve into humans, if you will, and their genome only changed 2% during those 8 million years. So we're 98% chimp. You can actually buy a t-shirt and wear it and display that. 8 million years. Poliovirus changes over 2% of its genome in 5 days as it travels from the mouth to the intestinal tract. So this emphasizes that with a lot of time, 8 million years, viruses can really do a lot.